cause too much injury and mask potentially useful genetic material. These nematodes were inoculated in celery cultivars 2 to 3 weeks after transplanting to allow sufficient development of roots and were maintained until harvest. Each treatment was replicated 3 times with 3 plants per replicate and arranged randomly in the greenhouse. The experimental plants were maintained at about 26 degrees Celsius under adequate moisture conditions. Data assessment. To determine whether the population of Meloidogyna incognita decreased or increased, 200 cubic meter of soil samples were obtained from each pat and processed using the modified Biermann tray technique. The nematodes were collected separately in 150 milliliter capacity Erlenmeyer flasks. The suspensions were allowed to stand at least 3 hours at room temperature, and the excess water in the flasks was reduced to 50 milliliter by suction using a pipette. The nematodes were transferred to 50 milliliter capacity test tubes and were again allowed to stand at least 1 hour for nematodes to settle at the bottom. The nematodes were counted under a stereoscopic microscope. Likewise, population counts of nematodes in feeding position were made from 1 gallon roots at random per treatment. Roots were stained in acid fuscin glycerol to facilitate counting. Data analysis. The data were analyzed statistically using analysis of variance of the Statistica package software. Homogeneity of the variances of the groups were tested using the Levene's test, while normal distribution of the dependent variable within groups was evaluated using the Shapiro-Wilks test. Differences among treatments were separated at 5% level of significance using Anne's significant difference test. Results and discussion. The growth factors such as plant height, root weight, weight of marketable and non-marketable yield were taken 8 and 16 weeks after inoculation. Percentage growth reduction was calculated based on the uninoculated control. The number of galls, egg masses, and the developing stages of the nematode, namely 2nd, 3rd, and 4th stage juveniles, and egg-laying females inside the roots and 2nd stage juvenile in the soil, counted per plant in the 2 assessment periods. Statistical analysis showed significant differences related to marketable and non-marketable yield 8 and 16 weeks after inoculation. A percentage yield reduction was obtained in tall Utah Supreme, and Utah 52 to 70R improved with 72.9 and 46.5% respectively 16 weeks after inoculation compared to plants harvested 8 weeks after inoculation. Non-marketable yield was higher in both cultivars 16 weeks after inoculation, while those plants harvested 8 weeks after inoculation was comparable with the uninoculated control. Plant height was reduced only in cultivar Utah 52 to 70R improved 8 weeks after inoculation, but not significantly different from the other cultivar in 2 assessment periods. Percentage reduction in root weight was noted in the cultivar tall Utah Supreme with 82% and Utah 52 to 70R improved with 18% 16 weeks after inoculation. No introduction in root weight was observed 8 weeks after inoculation. This study shows that response of Meloidogyna incognita differs significantly between celery cultivars and 2 assessment periods. Apparently, the effect of the root knot nematode to celery cultivars becomes more detrimental with increasing time. Significant reduction in yield, plant height, and root weight of these 2 cultivars was noted 16 weeks after inoculation. Similarly, the number of galls,